Well, I wouldn't say they were fun, but they were what I would call significant. We realized that calcium was involved in malign hyperthermia. And when I first started studying this, very little was known about what they call excitation contraction coupling mechanism, how stimulation of the nerve caused calcium to activate muscle. They even thought at one time that the calcium would be coming from outside the muscle cell and in. But then they realized that that wasn't the case. And George Palade, who won the Nobel Prize for identifying the endoplasmic reticulum in cells, led to the discovery of the sarcoplasmic reticulum in skeletal muscle. And then it was realized that that's where calcium was stored, in the sarcoplasmic reticulum. But then how was calcium released from the sarcoplasmic reticulum, and then what happened to it? And then there was first discovered a pump, a calcium pump, that pumped calcium into the sarcoplasmic reticulum. And so one of the first tests that I did was to try to determine if that pump was defective in MH. And as it turns out, it was not. And in fact, if you expose that pump to halothane, it actually increases the efficiency of the pump. So then I realized that it wasn't the uptake of calcium that was a problem, it must be the release. At that time, there was no mechanism for measuring calcium release from the sarcoplasmic reticulum. And so we finally worked out a mechanism for measuring calcium release, and it was calcium itself that induced the release of calcium from the isolated membranes that we had. And we were able to show that in the MH pig muscle that release of calcium was abnormal, and we realized that that was a calcium channel, which later on became identified as the ryanodine receptor calcium release channel.